Sorry. Oh, sorry. Voice is clear. Hello? Hello? Renuka Ashok.
Yeah, sir, clear, clear.
Right, right. So some disturbance. Okay, now clear. So yesterday we completed about the pteridophyta type studies such as lycopoding completed and equivalent completed, right? Hello?
Yes, sir, I am new to this class.
Yeah. So just compare with lycopodium and equisetum. So as I told you in during practical training classes, I clearly have explained clearly about bryophyta general character and pteridophyta general character with the comparison also. The same concept apply here. Then only you can learn easily any type study, whatever it may be, belongs to bryophyta or pteridophyta, whatever it may be. But you should learn the concept of general character. So anyway, as we learn yesterday, lycopodium and pteridophyta. First, we start with main plant body. What is main plant body in lycopodium or equisetum? Or of course, in today class, marsilia also. The main plant body called as uh, one second. Sorry for disturbance. So if you take any type study, right? Take any. Right. Roots and stem, leaf. Right. So these are the sporophytic plants, parts. That already you know how the roots here, the roots are the branched roots. Clearly we can see in diagram branched roots, you can see branched and adventitious roots. The function of root fixation and absorption of water. And what about stem? Stem is the horizontal like this, and this horizontal structure called as rhizome. Horizontal structure called as rhizome. So rhizome is present. From actually these rhizomes are consisting nodes with internodes, nodes and internodes. So from this region, for example, this is the node, and this is also node, this is also node, and this is also node. So write this. So this node between the place internode. This is the internode place, internode I, N node, N node. From each and every node, leaves are arranged. That is the next one, leaves. Next one, leaves. If you take the leaf structure, elongated petiole, you can see in diagram, elongated petiole. This is the petiole, elongated petiole, and a leaf. And you can see the leaf is the not a simple leaf, compound leaf. Compound leaf means it is more made up of more than one leaf. One, two, three, four leaflets. Four leaflets arranged. So that's why it is considered as a compound leaf. So the compound leaf is one of the advanced character. It is resembled to the angiospermic plant. So this is sporophytic plant body structure. And from the rhizome, the reproductive organs also developed. And in general, what are the reproductive organs in Pteridophyta? Respond anyone? Hello? Hello? In general, what are the reproductive organs? Reproductive part. Voice audible or not? Hello? Uh, hello? Voice audible or not? Tell me, please respond. My voice audible or not? Oh, where it is? Uh, please respond. Someone, Samsung, Rain Kashok. My voice audible or not? First of all. Hello?
Yeah, sir. Clear, clear.
Right, right, right. So and now tell me what is the reproductive part in Pteridophyta? I'm not asking about a particular Marsilia, Lycopodium, Equisetum, but in general, reproductive part.
Antheridium, Archegonium.
They also reproductive part, but I am asking about dominant stage. First of all, what is the dominant stage in Pteridophyta? Before that, you clarify me how many stages in plants in their life cycle. How many stages? I clearly tell you during practical training. And did you attend class, practical class? Renuka Ashok, did you attend or not?
Yes, sir. Attended someone.
Attend in botany or some other subject?
Yes, sir. Botany.
Right. Uh, actually, in botany lab, English medium student clearly explained about general character to avoid the time safe. So there, in what first of all, tell me what is the dominant stage, either sporophyte or gametophyte in Pteridophyta? Obviously, sporophyte. So sporophyte, of course you're right. Antheridium, Archegone, reproductive organs, right? No doubt at all. But I'm asking sporophyte reproductive part. What are produced as sporophytes in their life cycle, ending of life cycle? Hmm? Hello? Simple. Sporophyte name itself given. That's why it is produced the spores. So the spores, sporangia, sporophyll, commonly known as cone or strobila. Actually, the cone strobila is the excuse me, reproductive parts actually. But in Marsilia, it is somewhat complex. The complex structure called as sporocarp. You can see here. You can see here sporocarp. This is the reproductive part. You must have assumed what are present inside the sporocarp. Hello? Simple. Who is Samsung? SMX X205. Please respond whether it is right or wrong, no matter. Okay, sporocarp name itself given. Sporocarp produces the spores. Right. Spores. Anyway, I will explain in detail one by one. First, let's discuss about rhizome internal structure. This is important in theory as well as practical also. Very important for section cutting. And internal structure of petiole also. These two very important. The root internal structure not an important one. This is not important one. The important one only rhizome, internal, and what is that? And the petiole internal structure. And before that, in some general points of about the Marsilia, the Marsilia commonly known as pepperwort or water fern because they are present in water. That's why it is called a water fern. Um it is actually it is a fern, but it is oddly resembled to true fern. Why it is oddly resembled to true fern? Because it is already resembled to angiosperms. That's why it is oddly resembled to the true fern. It is represented about 53 species, which are cosmopolitan, can everywhere generate, can survive everywhere in distribution, and about abundantly found in tropical, mainly tropical countries like Africa and Australia. Almost nine species have been reported from India. Uh, these species based on the genus and species. Maybe they are hydrophytic or amphibians. Uh, that's it. So already discussed external characters also. Now internal character. Let's discuss first of all. Let's see the rhizome TS internal structure. Already told you if you take rhizome. Uh, did you take section from rhizomes? Was it given by sir yesterday? Hello.
Hello.
Was it given by yesterday? That means in my
No,
about
sir. I
what
have come.
what what did about the section cutting yesterday?
Sir, I did not completed a botany practical test.
You did not. Anyone completed? Hello, Samsung. You completed or not? Okay, leave it. But keep remember Renuka Ashok. In when you attend the practical training classes, just focus on Marsilia petiole and rhizome. These are the very important for practical as well as theory also. Okay,
Okay,
right.
sir.
Now root no need root transfer section of leaflet no need petiole no need circle outline and shows following structure if there is the outer stem rhizome character. All right. What is the internal parts? All I told you. Any parts internally mainly divided into three regions. What are those? So this is Marsilia transfer section. I tell me clearly. You can see the diagram. When you take a unit and make a section, you can clearly observe like this type of arrangement. So I will explain clearly if you where it is. You take, yeah. So in internal structure of rhizome, clearly three parts: epidermis, or like this common, cortex, this is also common, and the steel. Three region. But let's discuss one by one. Epidermis. Epidermis always uniseriate. You can see. Let's draw diagram here. Let's uh, where should I draw? So, for example, our convenience. Let's draw here. This is single layered epidermis. Sorry, my hands somewhat showering due to the fever. Uh, mm, this is epidermis. So this epidermis is single layered. And not only rhizomes, if you take stem and leaf and root, the always epidermis, limiting layer, single layer, thick parenchymatous, and stomata absent in epidermis. If it is leaf, stomata present. Of course, in some stem, stomata present, but root and rhizome. In some rhizome, also stomata present, but not here. Next, cortex. In the cortex, classified into three regions: outer cortex, middle cortex, inner cortex. Let's draw with different type of color combination. First of all, let me take blue color. One, uh, green. Sorry. So this also cortex layer, middle cortex and inner cortex. Inner cortex. Let's draw one by one here. First of all, this is epidermis clear. After below the epidermis, outer cortex. Next, middle cortex. In uh, inner cortex and last end with endodermis. This is inner cortex. Inner cortex. Let's discuss one by one. Uh, Here, outer cortex is present just below the epidermis and called hypodermis. Another name, hypodermis. It is parenchymatous. That means the entire cell region made up of parenchyma. Uh, Next to middle cortex, it is aerenchyma. Middle cortex, it is aerenchyma. That means the gap, air gaps are present. But it consists of large air space and air chamber separated by one cell thick parenchyma septa, xerophytic spaces, a chamber, and inner cortex, solid tissue, several cells, outer layers, thick and walled, sclerenchymatous. So this is sclerenchymatous. But remember, the middle one always air space like this, somewhat air space present. Right. Next, after inner cortex, last next one, cortex completed. Three parts completed. Next, stele. The stele is amphiploic cypnostele. This is important. Amphiploic cypnostele. What is amphi? What is meaning of amphi? Hello. Uh, uh, Here. Amphiploic cypnostele. This is very, very important. What is amphi means? Amphibians means. Hello. Amphi means two. Both. Amphibians
Snakes,
means
example snakes, amphibian.
right. That means it utilizes the two habitat for that survive. So if the amphi means also two, phloic means phloem. What are the two phloem? Actually, if you take the in already you must have learnt in lycopodium stem, ecosystem stem. Maybe inside the steel, outside phloem, inside xylem. That's it. That is the procedure. Most of the. But what happened in Marsilia rhizo? What happened in Marsilia rhizo? Both was actually middle xylem present. Let me change color. Right. This is X for xylem. Xylem present. Right. Now either side phloem. That means inside and outside. Another color. Outside phloem. Inside phloem. P for phloem. Like this. And also not uh, pericycle also to epidermis. Sorry, endodermis also two. So, but anyway, here the one very important one, amphiploic steel, and in the center is pith, which may be either parenchymatous or or uh, mostly parenchymatous or sclerenchymatous. Xylem is present in the form of complete ring. You can see in diagram, surrounded by both sides by completing inner and outer phloem, pericycle, and endodermis. Right. Inner phloem, outer phloem, inner pericycle, outer pericycle, inner endodermis, outer endodermis. But center region, medulla or pith is present. Center medulla, pith medulla, maybe uh, parenchyma or sclerenchymatous. Right? Any doubt in this? Hello? Hello? Right. Let's see the, you can see diagram also here. Uh, what I draw here, this what, let's change color, clear red. So red color, this region completely xylem, uh, inner phloem, outer phloem, like this. Actually, in diagram, they are mixed together. That's why for clear view, I had to draw like this. But anyway, keep remember this rough diagram. Based on this rough diagram, you can understand everything, right? Now let's move to the petiole TS. Now Marcella petiole. If you take same petiole, also here also same characters, but somewhat change. What are the same characters here? TS petioles over circular outline. Same here also same like rhizome, epidermis, cortex, stem. Always epidermis common. Outermost layer, single thickness, cells are parenchymatous, slightly elongated. No change, single layer. You can see it, the epidermis. No need to draw diagram already given here. How many layers? Single layer. Cortex, again, like rhizome, we are differentiated into three cortex. What are the three cortex here? Let's see. Outer cortex, middle cortex, and inner cortex. This is outer cortex. Hypodermis called as outer cortex. Present just below the epidermis is called hypodermis. It is made up of thin-walled parenchyma. The same character like rhizome. And middle character the same lies below the hypodermis and called erenchyma. You can see clearly diagram this erenchyma structure. With air, this is air filled with air, septa with air. So this all called, these all parenchyma cells filled with air. So erenchyma. Inner solid tissue, several thickness, and this is layers. Parenchymatous contain starch and tannins, filled cells. There are. So this is parenchymatous. Right. Now, steel. Actually, steel not clear here. So anyway, if I draw steel, only steel, because in this mixing you can't understand clearly. Only the epidermis you can understand clearly, and cortex you can, but not steel. But let's draw the steel here. Only steel region and arrangement of xylem. The triangular outline, proto steel type of pith. Remember, pith is absent. What is a rhizome? Rhizome with pith. Petiole, no pith or medulla. Xylem is V-shaped. Two distinct arms. Each arms provided metaxylem element in the center, and protoxylem is situated at the both margins. Protoxylem both margins. And protoxylem is exarch. Simple and simple. Protoxylem, that is protoxylem, small protoxylem. So this exarch outside, the small one outside. So that is exarch, and this is called V-shaped. So how many arcs like this? Two arcs. Also same V-shape. So this is protoxylem. This is metaxylem. Center metaxylem, outer protoxylem. So this V-shaped is the characteristic feature. Right? There is no pith. Is there any pith here? No pith. Only xylem present and surrounded by phloem cells. Either side. T for phloem cells. Here also same. We can draw here. Uh, right, like this. So keep the record and rest of the cells. This is phloem here. You can see here. Right. Is it clear? Any doubt in this? Allow, allow, allow. Are you there? iPhone, Renka, Samsung. Uh, right. Next, after internal structure, let's discuss about reproduction. What is reproductive parts? Already told you. Reproductive part is sporocarp. Let's see the structure of sporocarp. This is Marcellus sporocarp. Where this is, first of all, this is this sporocarp. If you take this sporocarp, vertical section appear like this. In this section, what we discussed here, let's see. No need about root internal structure, but only the important one is reproduction. Hmm. The section is, it is given inside horizontal longitudinal section as sporocarp. You can see in diagram also. Hmm. This is longitudinal section. Vertical longitudinal section. Right. So before that, you should learn about mm, one important thing, reproduction, origin of sporocarp. Maybe according to some scientists, nature of the sporocarp to main view have been put forward by different morphologists to explain the morphological nature of the sporocarp. One is first laminar of leaf segment hypothesis. Second, petiolar leaf hypothesis. Two. In first, the laminar of leaf segment hypothesis, according to the supporters, with this hypothesis, sporocarp is lateral or differentiated with given below. What they assumed, according to Bower, the hypothesis seems to have enabled that the sporocarp consists rachis, bearing row of pinnules. This is indicated by venation. That means it directly said the development of sporocarp by modification of leaf part. So leaf modified as is sporocarp according to some scientists, but some argue no, not it is leaf, only from the petiole region. From petiole region, rhizomes are raised. One another scientist only rhizome involved the formation of. So anyway, they are their different view as of opinions. They are proposed with come forward with their propositions. So anyway, whatever it may be, they evolved from leaf or petiole or rhizome, whatever it may be, the structure is same, never change. Here, excuse me. First of all, if you see in this longitudinal vertical section, first already told you the sporocarp produces the spores, right? But which spores? If you take lycopodium and equisetum, only homospores are produced, similar. But here, this is very, very important. Heterospores are produced. This is very, very important. Heterospores produced. That's why it consisting sporocarp, two sporangia. Inside the sporocarp, two sporangia. That is macro and micro. From macrospores, microsporangia, microspores. So these two considered as a heterosporous condition. This is called heterosporous condition. Let's see in this diagram. So in longitudinal section, clearly epidermis present outline protection layer epidermis and it attached with the stalk. Below the epidermis, hypodermis, clearly hypodermis. So this is considered as sporangium wall. If you see the structure, uh, let's draw here, clear view. Right. This is sporocarp wall, complete together sporocarp wall. Sporocarp wall, the first one, epidermis, this one, second layer below, hypodermis, clear. So these two together called as wall. The below the wall, gelatinous ring is present. This is gelatinous ring, continuation with gelatinous. Let's change color. Gelatinous color. This green one actually. This gelatinous ring. Right. This gelatinous ring also present. Uh, from the gelatinous ring, sorus arranged. This sorus protected by inducium. This is important. So let's change color. Blue. Let's draw blue. sorus, sorus. They are formed alternately. Year one and another axis year one sorus. Right. Alternately formed sorus. This is called sorus. Blue color sorus. Let's write sorus. In sorus, it is protected by inducium layer. And let's change color. Which color? Let's take red color. Sorus inducium. Protected by inducium layer. Right. This is inducium. Red color layer inducium. Now, what are present inside the placenta? This is placenta area. This is also placenta where the microsporangia, megasporangia arised. That place called the placenta. Placenta gives nutrition to developing mega and microsporangia. First, in this, let's draw with red color. Megasporangia, large one. Megasporangia. This is mega. Either side, let's change color. Um, Blue. I think red. Green color. Small one. Small means micro. Microsporangia. Both are present in same source with inducium. Right? Is this structure clear? Right? When reach maturity stage, automatically this inducium layer disappear. That means almost utilized to developing spores. Already you know what happened inside the megasporangium and microsporangium. Can you tell anyone? Hello? Megasporangium, what happened further? Hello? In megasporangium as well as microsporangium, meiosis division takes place. Due to meiosis division from megasporangium, megaspores, microsporangium, microspores. Microspores are released. But this is the very, very important thing is after releasing micro and megaspore, endosporic development. That means inside the spore, already explained during practical training classes. Inside the spore, gametophyte developed. I will explain in detail with the life cycle. Is it clear so far? Hello. So anyway, someone asked important question. We don't give any important questions. Please attend online classes. Mm, right. Is it visible to you? Somewhat enlarged in size. Of course. Clear or not? Hello. Anybody there? Right. Anyway. Oh, oh sorry. Excuse me. Right. Life cycle. How to learn life cycle? Uh, actually, when we start with types, when we start with this Marsilia with the sporophyte structure, right? This is sporophyte. Let's change color. Already clearly explained the sporophyte structure is diploid to n. Discuss about root system and rhizome system and sporocarp and leaf, whatever it may be, and internal parts also clearly mentioned. I've just focused on the sporocarp. In sporocarp also clearly mentioned. Already told you two sporangia. This is mega. This is microsporangia. Sorry, megasporangium, microsporangium. These are from this megaspore, microspore develop. Inside, see, don't get confused. This megaspore is haploid N. Inside megaspore, developing the number of cells are formed like this. These are called archisporial cells. In this archisporial cell, one is developed as a megaspore mother cell. This one. Later it divides and forms the megaspore. These are called megaspores. Later this megaspore further developed into single cell egg cell. That means rest of actually divided, destroyed, only one remain. This remain one divide and redivide and forms some different type of cells. So in this most important one is the egg cell, female gametophyte or egg cell. Where is female gametophyte cell? Inside the megaspore. It not released out. In either way, here also inside the spore. Inside the spore itself. Divisions, further divisions takes place, number of cells are formed. This will further division and this is male gametophyte. This is also male gametophyte. How many male gametophyte developed inside the spore? Microspore, two male gametophyte. From each and every male gametophyte, male gametes are released out. Male gametes are released out. During fertilization, what happens? This male gamete with cilia, swim in cilia itself, meaning is with swim in water and fuse with egg cell. What happened automatically here? The zygote divided and formed the embryo. This embryo further developed into sporocarp. That's it. So this is development endosporic, not outside. When you see equisetum lycopodium, gametophyte can visible outside, but not in Marsilia, not developed outside, it developed inside the spore. So that is called endosporic. So this endosporic, this endosporic leads towards formation of gymnosperms. In all gymnosperms, the similar life cycle occurs. So this is advanced type of characters of Marsilia. Any doubt in this? Hello? Anybody there? Please draw this life cycle. It is very, very important. Even if you just write the names also. Names means, for example, this is sporophyte. Already told you, and again I will explain the next batch also. Diploid sporophytes root. I will do. Don't worry. Again explain root to stem, leaf, and the sporocarp like this. Sporocarp from monocarp, megaspore, microspore, megaspore, male gametophyte, female gametophyte. Sorry, this is female gametophyte, male gametophyte, male gamete, female gamete, here male gamete, like this. So just write the name instead of diagram. You can write the names also. That much also enough. Okay. Right. Any doubts? Any query in this? Anybody? Please respond. Hello. And this heterosporic condition leads to the seed habit, seed formation. This is also important in theory, not practical. We will discuss in detail tomorrow, next class, about heterospory. Already told you what is hetero. Hetero means different. Spores. The different means one is small, micro, large, mega. So what happened later? While they are formed as a micro and mega, automatically what happened? This megaspore, your embryo. This embryo further develop and instead of directly formation sporophyte, first it is covered with seed coat. Where is embryo? Inside the seed. Inside the seed embryo. And covered by seed coat. Like the seeds are formed. So this is leads to the formation of seeds in gymnosperms and angiosperms. So that's why this is very important. Heterospory and seed habit also important. And another important question here, stellar evolution. In stellar evolution, already told you protostele, you know already. You should write protostele. And ecosystem stele also, you know, that is siphonostele, advanced. And amphiploic stele also, you know, amphiploic stele and petiole of Marsilia. So this much enough in stellar evolution. These are very, very important. In Pteridophyta black, yesterday we completed Lycopodium, cone important, mainly cone for short answer. Ecosystem cone and stem. Lycopodium also cone and stem. Sorry, I forgot stem internal structure. Ecosystem cone and stem internal structure. In Marsilia, sporocarp, both internal structure, both means petiole and rhizome. This all together, that means all internal structure together develop as a stellar evolution. The stellar evolution also important. My lecturers clearly explain about the seed habit and heterospory. This is also very, very important. Seed habit and heterospory also important. Right. Any questions today? Hello? Any remaining in this? So if I complete the heterospory seed habit, stellar evolution both, the complete this complete pteridophyta completed. And next one, gymnosperm character. What are gymnosperm? Just compare with the pteridophyta. What are gymnosperms? Gymnosperms same like a bryophyta, pteridophyta, like bryophyta, embryophytic. First of all, sorry, I forgot. Autotrophic, embryophytic, like pteridophyta, tracheophytic, seed-bearing plants. Seeds are formed only in gymnosperms, but not fruit. These are called naked seeds. If you see sporophyte structure also, it is clearly advanced one. Clear, well-developed root, elongated shoot with branches, multiple branches, and different type of leaf. Of course, like Pteridophyta, here also the reproductive part is cone. But this cone is very complex. It is not unlike Equisetum and like Lycopodium. Complex cone present. We will explain later about gymnosperms, type studies, about Pinus and Netum. These two also important. In especially in Pinus, already you must have learnt about Pinus needle internal structure. This is useful for both theory and practical, as well as the Netum stem TS also. It would be useful to you both theory and practical. This much important. So please focus on gymnospermic character first, general character. After that, enter into the type studies. You don't worry. I will first of all I will clearly explain during practical training classes about general character. After that, we will enter into the type study about Pinus. We will discuss Pinus clearly and Netum clearly. This much enough. Right. Thank you. Thank you all. Next class, be ready with heterospory seed habit and stellar evolution, fineness, and neat. Right. Thank you all.